She graduated from Moscow State University, got his PhD in Paris and in in Montreal. Now he's the head of the department and work in bioinformatics, and was awarded two very prestigious prizes by Mathematical Classification Society. So, Vladimir, the floor is yours. Please.
Thank you very much, Fuad Tagevich. It was a real pleasure to respond to the invitation of Fuad Aliskerov during your summer school. In fact, I am not the director of the department. I am the director of a bioinformatics program in the Department of Computer Science at the University of Quebec in Montreal. And I would like to present our works in the field, mainly in the field of bioinformatics, but also I will talk about clustering. I know that Boris Mirkin will present tomorrow, and he is a specialist, much better specialist than me in the field. But clustering is my second passion with bioinformatics. So I will talk a little bit about application of bioinformatics in the field of clustering. I will start with recalling some basic definitions of bioinformatics, and then I will present our methods and also a web server called T-Rex that allows one to infer evolutionary trees and visualize them, compare them, and it includes some algorithms developed by our research group. So what is bioinformatics? Bioinformatics is a field that defines a set of methods and programs and online applications that allow one to manage, manipulate, and analyze biological and biomedical data. In resume, we have problems that are posed by biology and medicine, and we use computer science, mathematics, and statistics to solve these problems. What are the main goals, main objectives of bioinformatics? The main objectives, first is collect and store genetic and genomic information in databases, which are accessible online. Here we have a graph that presents the milestones of collection information, and we can have that it's exponential, the number of sequences, which are genetic and genomic sequences, which are now stored in databases, mainly in GenBank and in CBI, but there are other databases which are popular also. And also we can see the growth of the number of users. It's proportional to the growth of the number of sequences. So there is a huge interest for this genetic and genomic information, and some important dates in the field we can, for example, see in 2001 it was the sequencing of the first human genome. It was the milestone when the first genome of a human being was stored in the database. But now we have millions of genomes of human beings. The price of sequencing went down, and now we have the information about almost all viruses, bacteria, all species that that exist around the world, and that we can sequence. What are the main tasks that we have? We mainly work with sequences, and we need to compare them. And the main task when we have two or more sequences is to align them, putting in correspondence the sites of these sequences, aligned in order to compare them, to see where the mutations on some other evolutionary events has happened. What's important, we need to identify sequences in the database, and there are many tools in this database, especially in GenBank, it allows you to do it. We need to determine a similarity degree between 2 or more sequences. It's in order to represent them, their evolution, for example, to see how the sequences evolved, what was the evolutionary history of the species involved in this data set. Also, we need to find structural motifs, for example, genes promoters for nucleotide sequences, and for example, folding zones and active sites for proteins. Because we have from one part DNA information or RNA information stored in the database, but we have also amino acids or proteins which are also stored in this database. What another physiological and functional prediction can be done with by identifying operons. So it's genes having similar function. We can identify abnormal genes, predict 3-dimensional structures of proteins, and so on. And we can also conduct experimental prediction whose goals are to identify restriction sites, predict the digestion of nucleotides, and so on. Now I will go to the part that to the subfield, I would say, of bioinformatics in which I worked and I continue to work. It's the reconstruction of evolutionary or so-called phylogenetic trees. Here the idea is to represent having, for example, some genetic or genomic information. Is to draw a phylogeny or a tree that represents the evolution of the species at hand. For example, you have usually represented as 3 difference between these phylogenies. The first phylogeny, it's look like the other species, and in the second phylogeny, whale is close to hippopotamus. One could maybe in fact, the most logical explanation that the first phylogeny is the right one, but in fact, genetic information says us that the right phylogeny is the second one, and the second phylogeny in which whale is located near hippopotamus. And it's not only morphological features and how the species looks like that is counted here, but mainly the genomic information. We align 2 genomes and we compare the corresponding genes, and it tells us the genetic information for whale, that whale is much more close to hippopotamus than to other species. So the phylogeny on the right side is the right one. So the phylogeny or phylogenetic tree represents, in fact, it represents a history of evolution of given group of species. And often we work with molecular phylogeny that proceeds by comparison of genes, and in this comparison, we are looking for mutations. Mutations that when one nucleotide or one protein changes. Usually we work with nucleotides, and when one nucleotide changes, it may be considered. In fact, one nucleotide is not, maybe it's not enough, but several nucleotides, it may be enough for the formation of a new species. I will continue to explain how we proceed, how we infer phylogenetic trees. For example, on the right part, we have a representation of the parsimony method when on the root we have a species which is represented by a sequence of AAA. And after one mutation, for example, here, if A changes for J, it will allow us to create one species. We put 1 here. It means that was one change during this branch. A branch usually represents an evolutionary process, during which we can we can count some mutations, some other biological events such as recombination, horizontal gene transfers, and so on. And the ancestors of species are represented by internal nodes. The contemporary species are usually represented as leaves, and usually have a root, and the root is the most it's a common ancestor of all species presented in the tree. And here, by proceeding by one change every time, we can arrive to the species which is represented by the leaves. It's orangutan, gorilla, chimpanzee, and human. So it's an example how, but an artificial one, how some mutations can allow us to follow the tree reconstruction. In fact, what we have as input of most of the methods is the information related to the leaf. So we have those sequences, and the goal is to reconstruct the tree, to reconstruct the, if possible, the ancestral nodes, including the root. Also, we have another example here. It's if we have sequences S1, S2, S3, we can align them, putting in correspondence the corresponding site, and we can just use the information which is well conserved. Usually, what we do is this, and we can cut the second part of the line. Okay, so here we have a tree which is represented by its distance matrix. Usually we work with trees, as I said, whose leaves represent species, but the branches represent the number of mutations, but also the property of additive or phylogenetic trees and the sum of the lengths, which length is located on a path. There is a unique path in a tree. Always there is a unique path. Some of the branches can be counted and can be stored in as a distance matrix. Can be stored as a distance matrix. For example, here we have species B and A. The distance between them is 5. It's the sum of the branches 1 plus 4 of the unique paths connecting species A and B. Also, we can take another example. For example, between C and...
Volodya, sorry, just a question.
Yes.
If you don't mind, usually we can interrupt speakers.
Yeah, sure.
But these very distances, do they mean something? For instance, if the distance between F and B is 11, this means that they are not in the same group or something. Something separates them. You see, for instance, 7 is okay. There might be relatives, but 11 is too much. Do you make such kind of conclusions when you consider such distances?
In fact, each distance it represents it's kind of genetic distance.
Yeah, I understand that it's genetic distance, but is there some kind of threshold, for instance, which says that if distance is higher than 5, it is there are different spaces, etc.
Usually, when we reconstruct phylogenetic trees, we don't have thresholds, that will say it may be a good idea, but usually we don't consider thresholds that will say that if the distance is greater than 5, it's considered too much, or if less than 5, that species are closely related. Consider this, but species that we have can separate them only in one tree. It will be only very small distance. It can be also good We don't have the transfer here, a neighbor joining method or any other algorithm that will allow us to transform these distances into a tree metric. And this tree metric is representable by a tree. But if you have, it's a very good large distances in species doesn't really relate it to the other species or to the group of species. Maybe it can be cut or represented in another. I will not show how we can do it, but I think we can use a kind of clustering and cut the trees on certain level. I will continue with bioinformatics and what are the main tools that we use. For example, now we have a lot of applications for smartphones that will allow you to give information, several sort of information about your genome. If you now the cost of genome sequences it dropped drastically. It was $95 million in 2001 when the first genome was sequenced, and now it's around $500. So everyone can sequence his own genome and then use an application that will tell you what are potential diseases or RHs can be detected during the whole genome analysis. For example, establish the diagnosis for rare diseases, or it will even allow you in some cases conduct genetic tests, for example, those for cancer risk. So you can, it's not for sure the final diagnosis, but it can give you some preliminary information, and you may want to go to see your doctor if the application tells you that you have a very high risk of cancer in the future. We have these applications now more and more for smartphones, and it's used more. Okay, now I will show you. I'm working in the bioinformatics lab and show you some programs that we create and web servers that we created. I will try to show the slides, and I will now I will try to show in real time how the application work. I will start with the bioinformatics, direct bioinformatics server. Here on the slides, I will show some screenshots from our server. It mainly includes the methods for reconstruction and visualization of phylogenetic trees. We have several interactive manipulation tools for phylogenetic trees, and several views which are possible for phylogenetic trees, for example, radial, axial, hierarchical, vertical, or horizontal. And it's used by many researchers around the world. The paper about this web server received several hundreds of citations. In fact, why we developed this web server with this application, it's not only to serve the scientific community, but also to present our own method. Because with some standard methods, we are also included in the application in fact, almost all our methods in the field of phylogenetic analysis are included in the application, and it's a very good way to disseminate, to show our algorithm methods and results. It also increases the number of citations of our papers, and it's what I recommend to all my students. So always when somebody proposed a new method, new algorithm, create a software for this algorithm and make it available to the scientific community. It will increase the visibility of your work. Some other here are the examples of some pictures, some phylogenetic trees, and also networks that will can be drawn, can be analyzed using our tool. Here we have the interface for the selection of methods. We have distance-based methods that proceed by working with distances, with matrices, and we have also the parsimony maximal likelihood methods that use that work with sequences directly. They reconstruct trees from sequence without computing this. Also, we have our old work with my supervisor, postdoctoral supervisor, Pierre Legendre from University of Montreal. It's a kind of what we call reticulogram distance. So we first we reconstruct the phylogenetic trees, and then to these trees we add some extra branches, creating a phylogenetic network. Here we have, in this example, the vertebrate data set of 9 species, to which we added using a certain criterion. In fact, we used either least squares or weighted least squares criteria. We can add some extra branches. Here the extra branches are shown in purple. And what does it mean, these extra branches? These extra branches means that, for example, a bird, it doesn't mean that bird is genetically related to mammal, but it means that the tree model, because the tree, what we have, the backbone model, the tree model, it doesn't represent well this distance between bird and mammal, because in fact this distance is closer and it should be represented by an extra branch. The same is for lizard and turtles. They are more closely genetically related than in represented by the tree model, because each model is a phylogenetic tree. It's a model of a history of this species, and it's a kind of approximation of this history. We never know the real history. Another kind of application that we have, on which I worked a lot, and I believe Boris Мячин also worked a lot. It's how to reconstruct, how to detect horizontal gene transfer. How to detect horizontal gene transfer affecting certain species. Horizontal gene transfers are especially important and influence the evolution of prokaryotes, of bacteria, and also the evolution of viruses. So when we deal with this, with the evolution of these species, we shouldn't only represent this evolution by phylogenetic tree, but there are some other evolutionary events, such as horizontal gene transfer and recombination, which are very, very frequent among bacteria and viruses, which should be represented as a phylogenetic network. In this kind, in the case that I show, we have a directed network, because here we have directions on all the branches, on all extra branches. We have the tree, and after it, in this tree, we map the extra branches that represent horizontal gene transfer between the related species. And horizontal gene transfer, it's a directed event. So for this kind of information, we need to use, to represent this kind of information, we need to use phylogenetic networks which are direct. We have some other useful phylogenetic application in on our servers, which I will show you right now. Among them, we have some applications for aligning sequences, either two sequences or groups of sequences, and for computing different kinds of phylogenetic distances, which is also always useful when you have sequences and you want to translate them into distances. There are several ways of doing so. Maybe I will now show an application of our methods, for example, for inferring horizontal gene transfer and recombination, how we apply them on a large scale to analyze the evolution of prokaryotes. Prokaryotes are species that don't have nucleus, and it's mainly bacteria and archaebacteria. As I just said, there are several evolutionary mechanisms that cannot be represented by a tree. And for example, the evolution of bacteria and viruses includes the evolutionary mechanisms such as recombination, causational gene transfer, and this mechanism should be represented by a network. And there are some other evolutionary mechanisms that require phylogenetic networks. These mechanisms include species hybridization, recombination, homoplasy or gene convergence, and also gene duplication followed by gene loss. All these mechanisms should be represented by phylogenetic network. Our main method for inferring phylogenetic trees, it proceeds by, usually we work with two trees. One tree which represents the species evolution or genome evolution. We can take this tree from the gene bank, for example, database. And we have a gene tree also which represents the evolution of one particular gene. And we want to see if this evolution matches the evolution of the whole genome, whole genomes for the same species. And if the phylogenetic trees are different, we infer two trees, and if these trees are different, we need to proceed by reconciliation. Reconciliation of these trees. I will explain you how it works. What we are doing, we need to proceed by subtree moves, allowing us to transfer the species tree into the gene tree. We fix the gene tree topology; it doesn't move. But in species tree topology, we will cut some branches and grab them, put them in a different place in the tree in order to match the gene tree topology. We need also to incorporate necessary evolutionary constraints in our algorithm, because, for example, some horizontal gene transfer within the same lineages should be prohibited. We cannot allow horizontal gene transfer between ancestors and predecessors and descendants, for example. We also have to maintain time complexity polynomial, because the general problem SPR of convenience SPR distance is known to be NP hard. And if you work with trees with thousands of species, the time complexity of the method should be polynomial. And what we also proposed is to a kind of bootstrap mechanism that will allow us to assess statistical support of the obtained reticulations branches, of the obtained horizontal gene transfer. How it works in practice with a small example, we always understand better with a small example how a certain new method works. Here we have a species tree on the left and gene tree on the right, and the idea is to transform the topology of the species tree in order to obtain the gene tree. The branch length here doesn't count. What is important is the topology. So we can see, for example, is here. Okay, the first transfer that can be done, it's transfer from species A to species D. It will bring, it still is in the species tree, and it will bring the species D closer to the species A. And this way, these two species will be located as it's done in the gene tree topology. So this is the result of this operation. So we cut the branch of D and we move it to the species closer to the species A. So A and D will be now neighbors, and D will be cut from its old location. The second transfer that we can do here, it's from E to B. It will cut the species B and bring it closer to E. It's how the tree will look like after this operation. And the third horizontal gene transfer that we need to do is from C to F. What it will do, it will bring the species F closer to the species C. Here is the result of this operation. And in the end, we obtain a tree topology which is exactly identical to the gene tree topology. Okay, here is the solution, and the solution, it will be a network. It's a species tree with some extra branches. Here we have 3 directed branches because we did 3 gene transfers, and the solution will be a phylogenetic network, a directed network, because this the direction here is important. I don't know if you have questions. No?
May I ask a question? Is it
Yeah, sure.
has it applied to real data, to kind of with biologists? Did you do any applied work with this?
Yeah, sure. We applied this the algorithm that we created in our lab. We confirmed with biologists. You know, it's in fact our bioinformatics team proposed the algorithms, but after it we applied this algorithm on real data, and we'll just show an example of such an application. And the biologists then interpreted and confirmed all our results. It has been done, yeah. Question. In fact, in bioinformatics, we have no choice. If we propose a new method, it should be always confirmed and confirmed by biologists.
At least checked, yes.
Yeah, exactly. Because we can propose something, it can work theoretically, but in practice, when we apply it, because with this method, what we proposed first, the method worked pretty well on some theoretical data, some artificial data, but when we applied it on real data, we understood that it works only for trees of size 10 or less. And if we have trees of 100, 1,000 species, it doesn't work because the time complexity is so, it was huge. But we were almost able to find the exact solution, the optimal solution, but we didn't consider that the time complexity is also very important. And the time of the running time of all methods proposed in bioinformatics, it's a huge point. It's a huge challenge, and we need to take it into account. It should be a good heuristic, because most of the problems are NP hard. But what we need to propose from one side, we should provide a good solution which is close to an optimal one. But on the other side, we should do it in a realistic time. So I will now show one example of application. Here we considered the different families, I believe 23 families of prokaryotes. It's bacteria, different bacteria and archaeobacteria, all bacteria. And the idea was to check what are the main gene exchanges between these families. And what we did, we applied on a large scale our horizontal gene transfer methods. And in fact, there were 2 methods also. One of them allowed us to infer complete transfers when a gene is transferred and it's incorporated in the whole genome without changing something in it. So we just add the gene, and this gene will replace the analogous gene of the whole genome. And the second kind of methods, also in the field of horizontal gene transfer detection, it was the methods that work with partial horizontal gene transfer. Why I call it partial? Because the horizontal gene transfer was followed by recombination, and the transferred gene recombined with the homologous with analogous gene in the host genome, and it was creating a kind of mosaic gene. How we call it, and the mosaic gene, it contains parts from both the host genome and from the genome that the donor genome. So, and it was another challenge. It was much harder to detect just a transfer that doesn't provide any mosaics in the gene, and we had to separate. We had to separate these two analyses because the methods were completely different. Now what we obtained? After the application of these genomes, of these methods on the genomes of bacteria and archaea. Both of them are proteobacteria. And this is a heat map from the complete horizontal gene transfers. We can see the families between which we have the high transfer, and we can see that in most of the cases we obtained very low results of gene transfer or complete gene transfers. In fact, the numbers that we have here is the probability that a certain species from one particular family was affected by horizontal gene transfer from the other family. For example, here the highest values were obtained here with 7 and 8%. It means that the species from other archaea were affected by Nana archaeota and Crena archaeota. It was the highest numbers that we obtained. Now I will move on. Okay, and now we did the same analysis with partial horizontal gene transfers. Here we were looking for mosaic genes, and here we can see that the heat map contains much more different values, and we can go up to 100%. And we can see that the mosaicism and the creation of mosaic genes is a very frequent event in biology. And here the numbers are much higher, as I said, and we also have a heat map that was analyzed by biologists, and this is something that some results that we know have not been obtained or shown, cannot be found in the literature. So I hope to publish it soon. And at the same time, when working with the same gene families, we represented our results by habitat. Habitats, it means that we look at this time at the habitat where these particular families or this particular species live. And here we can also do the same analysis with complete gene transfers and partial gene transfers. For example, for complete gene transfers, we obtain here this kind of heat map, and we can see between which habitat we have the highest gene exchange. For example, for human others, we have most of the transfers comes either from human others or from plants or from animals or from soil. The same can be done here for mosaic genes or partial horizontal gene transfers. And the map here has much more colors. It means that the exchange was much more intense. And also we can see that human, for example, human respiratory, most of bacteria comes from another human respiratory or from air or soil. For example, for human other, from some intestinal, for example, bacteria, most of them comes from another intestinal bacteria or from plants, from animal or from soil. And so on. So this analysis can be done not only on phylogenetic gene families, but also within a habitat. And the result needs to be interpreted, yet. So we are on the way of doing so. Maybe what I can do now it's to show you the web server. I will switch to the to my web browser. Can you see my web browser? Maybe it takes some time.
Yes, we can see. Yes.
Oh, okay, so perfect. So I will show you how this method, some of this method works in practice. So what can we do? First of all, I will show you. Now I will clear all this. We can draw some phylogenetic trees. This. And then we can save them in a certain format. Here we use a Nuix string format. Oh, so it doesn't work. If some operations were not good, I will do something simpler. Okay, this tree should work. So we can encode this tree into so-called Nuix string, and then come back here and decode this string using another application. Convert it into distance matrix, and then we can take this, copy this distance matrix, and use any method of phylogenetic tree reconstruction from distances and compute a phylogenetic tree. So it's going to work. No, it doesn't. Okay. I need to indicate that we work with distance data. Compute. Okay, now it works. And here is a tree. Here is a tree that we obtained. Okay, I will show you some other applications. For example, tree viewer will allow you with phylogenetic tree dataset still you to different types of tree drawing. For example, we can there are some trees that can have roots, branch length specified or not. It can be trees with bootstrap scores specified, bootstrap scores that represent statistical support of tree branches or not. And all this can be visualized using our application. For example, this tree, I will try to change the type of drawing. Okay, so this is the hierarchical vertical types of drawing. We can also have the actual type of drawing where the tree is located near, it's located along its longest axis, which is the longest path in the tree, which is this one between salamander and turtle. Also, we can have this type of radial drawing when the tree is located within an imaginable circle. So it's located via a circle, and it's pretty useful for copying it. We can copy it and put in any of our articles, and it became really popular among the scientific community nowadays. So now I will show how the phylogenetic tree reconstruction methods works. We'll use this vertebrate data set. For example, here we have the data matrix, and we want to infer a tree. Okay, click on compute, and what we have the tree. Tree is here. The tree is in Nuix format. Here. And we also have a page of statistics, which are important. For example, the tree metric or additive distance matrix inferred from the input basis matrix is as follows. So we also have some useful coefficients that we compute. That, for example, is squares coefficient for this approximation, and some other coefficients. The total length of the tree is also important because it represents a kind of way of evolution. We try to minimize the total length of the tree because it minimizes the number of evolutionary events that has happened during the evolution. And also the representation of tree as a list of tree branches with their length. Back to the main menu. Now, what we have here, we have also methods that allows us to reconstruct trees from incomplete distance matrices. So in some cases, we cannot measure distance between genes or genomes ahead. And here we replace these values by -99. There are several of them here in this example distance matrix. And there are specific, because the classical method doesn't work in this case, and we need to apply some special methods. And one of them is here. It's one which I proposed a long time ago with François-Joseph Lapointe from University of Montreal. And this one works and also provides the tree. Okay, the tree is like this. In the tree, we shouldn't have any missing value, but in the original distance matrix, we had some missing value. So, back. Okay, now reticulogram inference. Go back to the vertebrate data set. Here we can, it's a method that I also explained before. Also, the input is a distance matrix, and the output is a reticulogram or an undirected network. Here we can have some extra relationship between species compared to the phylogenetic tree model. Compute. Work. Okay, here is the result. The reticulogram, in fact, is a tree with one extra branch between mammal and bird that shows that the relationship between these two species are much more close than it's represented by the classical phylogenetic tree model. Okay. We also have some statistics here. And we have some criteria which we call Q1 and Q2 for adding these as extra branches. The criteria are based on either least squares or weighted least squares. Okay, now I will show some methods for detection of horizontal gene transfers. Here I will take one example of real data. For this, we need two trees as input, the gene tree and the species tree, and we are trying to reconcile them using our algorithm. And what it should give in the end, it's a kind of directed phylogenetic network. Compute. Okay, so the network which we obtained for this example, in fact, it's a species tree plus 5 extra branches, directed branches, which represent horizontal gene transfer between the species involved. Here we have 5 of them. We also stop after a certain criteria, after a certain condition, a certain criterion is satisfied. What would be interesting? For example, we can also, as I said, one of the basic operations in bioinformatics is to compare and align gene or genome sequences. Here we take the data, it's a protein data or amino acids data for 8 different species. It's for 8, no, for more. It's for 11 species. It's encoded in the FASTA format, and the idea is to put in correspondence the right sites, the corresponding sites, and let us align these sequences. Here's the input file. We have, and what is it? Is the output file. Here are the aligned sequences. So we can see that the corresponding sites are in the right order. Also, we have this a kind of tire symbol. It represents some missing information, missing symbol that can be missing symbols that can be present in some sequence. I'll come back. Also, we have two other applications. I can show them to you briefly. One of them is Armadillo. This one, it's an application that will allow to do a kind of complex phylogenetic analysis or complex bioinformatic analysis using workflows. So we can create a kind of workflow to draw it on a chart and then proceed by executing all these methods in a line. It will create a kind of complex pipeline which we will execute, and this type of tools is useful when we do the same analysis, same operation many times, when we analyze the data using the same bioinformatic tools. Here's an example of complex workflows which can be created using Armadillo. It was done with, especially with my PhD student, Etienne Lord. And another one, which is much more recent, it's Simplot++. We call it like this. It's a software that allows us to, in fact, to represent, to visualize first, and also to compare sequences graphically. Here, there is an example how we can compare the sequences. It's by drawing a similar sequence on the same plot, and by this kind of plots, it's called similarity plots. And what we are measuring here is the sequence similarity. Usually we have a reference sequence and a bunch of sequences to which we want to compare this reference sequence, and we create a kind of plot, usually using different distances. We can do it, and it provides information where the sequences are closed, where there is a difference, and it's especially useful for analyzing some virus and some bacterial data. And here we have different types of analysis, including the network analysis. I believe it's here. Okay. Okay, sequence. We can create a sequence similarity networks that show the transfer.
So you said network analysis. What kind of networks you consider?
Okay, here the network that we consider is undirected network, and the network nodes represent species, and we can call it a sequence match between two nodes or between two sequences if they are similar enough. We select a threshold, and for this threshold we create a graph. It may be complete graph, but usually it's kind of graph in which sequence similarities are shown by edges. So we have an edge between two vertices if the similarity between these two vertices or these two species is high enough. If it's higher
then I have a question. Is it not possible to put some numbers on these edges measuring similarity? For instance, these two vertices are similar in 0.5. Those two vertices are similar as 0.8, for instance.
Yeah. You see on the. I hope you can see. You can see my screen. Here we have especially two different thresholds, and two different thresholds. One it was a threshold for complete gene transfers, and another one for partial gene transfer. And for each of these thresholds, we have different colors of edges. For example, the red one it's for partial transfers, I believe, and for complete transfers it's black lines. So it may be two different thresholds that can be used, for example. And yeah, I know that the networks are well used in many fields, in social analysis, but they can be used. It's our new software. We also insist that they can be used in bioinformatics, in the phylogenetic analysis, in order to represent gene transfers. In fact, gene transfer, it's a kind of link between two elements. If for example, two species, two people collaborate, we represent by them by a link. And then we can analyze these networks using different tools, like tools for doing so. We can try to find the central elements. We can try to find the, for example, species or bacteria that gives the most transfer to the other species. The bacteria that receives most transfers of some different species. And there are some different analyses that can be conducted related to the network. Okay, thank you for your question. I will move on. I will come back to my presentation. PowerPoint presentation. Okay. What I can show here also, it's the second part of the analysis where, okay, I will come back to the full screen mode. It takes time to find the right slide. Okay, we also applied this horizontal gene transfer and combination analysis to analyze the beta coronavirus datasets. It's related to SARS-CoV and the SARS-CoV pandemic. What was found in the literature? It's in fact that for some genes, the genome of SARS-CoV-2, some genes of SARS-CoV-2 are much closer related to the bad genes, but for other genes, it was found by genetic analysis. It was originally found by Chinese researchers. It's related to the species called pangolin. So we some SARS-CoV-2 elements, beta coronavirus elements, were found in the genes of pangolins. They were also found in the genes of bats. For example, the species rat J13 was used, and the question was how it could recombine in order to create a SARS-CoV-2 genome that we have now in practice here. That was the origin of the pandemic in 2020. And here we conducted some analysis. First of all, we inferred the phylogenetic tree, and there are here we considered 4 human species of SARS-CoV-2. It's here on the top part of the tree, and we also considered some beta coronavirus species found in pangolins. They are in red here, and all the rest it's the beta coronavirus species found in bats. The tree is like this. The tree is like this. So we can see that the closest neighbor of the human SARS-CoV-2 genome, it's rat TJ13, which is a bat coronavirus. But for some parts of the genome, it's a similarity analysis to be conducted. It was not the case. Here it's we found that, for example, for gene S, there are 11 genes in total in the SARS-CoV-2 genome. For gene S, there are some parts of it which is much more close to the beta coronavirus of pangolins. So we conducted some analysis using fit test about the recombination, and we detected recombination in here in seven genes of out of 11. That means that horizontal gene transfer and recombination affected this coronavirus genome. And these genes were found mosaic, and in fact the parts of the genes came from different species, different bat and pangolin species. What we found, and what we in the papers that I published with Pierre Legendre and some of my students, we were able to conduct this horizontal gene transfer analysis, and we also found the concrete horizontal gene transfers, and we represented them as a network, as a directed network. And we also found the exact locations of these transfers in the tree and the exact locations in the genome. It's represented on the here for the gene as on the right part of the tree, because what we found that some of the genes were affected by transfer from pangolin and some from different species of bats. And the same was done for the RB domain. It's a small domain where which is responsible of the gene S, which is responsible for the penetration of the virus to the host genome. And here we found this famous transfer from pangolin to the human SARS-CoV genome. Here are the main gene transfers that we found. The final diagram is different gene transfers represented by different colors, because it's coming from different places, and also we use different colors to represent either complete transfers or partial. Here are some references. Maybe in the ongoing project, it's not related to bioinformatics, but it may be of interest to most of you. Okay, it's what we are actually doing, and it's closely related to decision making analysis, and to the decision making process. What we are actually trying to develop, it's an automated recommendation system based on deep learning, and this system, what it will allow us to do, it's first to find the most appropriate clustering algorithm for a given data set. I come back to clustering. B, it will allow us to find the optimal number of clusters for a given data set. The given data set is the input of our system. And then we also will try to find this clustering. So this clustering, it means the separation of a given set of elements into clusters, into closed groups, according to a certain criterion. Usually we use a similarity criterion or a kind of distance. For example, it can be a Euclidean distance, it can be Minkowski distance, or any other distance can be used as a criterion for separating the elements. And we are trying to accommodate, to put the closest elements to the same cluster. Usually it's done now using, there are different cluster validity indices. Boris knows them much better than me, such as Kalinsky-Arabat, Silhouette, and others that are used first to find the optimal number of clusters in a dataset, or to find the most appropriate clustering algorithm. But we can show that these indices are not optimal. They are not optimal, and we can propose a kind of general recommendation system that can beat them. How the clustering works, just this slide shows that there are different kinds of clusters, and some methods are much more appropriate for some types of cluster. On the case A, we have a kind of circle clusters where we should use density-based clustering algorithms. The same density base should be used for these banana-like clusters or moon clusters. But if we have some clusters as case E, which represent Gaussian circles or ellipses, some methods like k-means can be used, which are very popular and very fast. And we can have different kinds of overlap. For Tagich, you have a question?
No, just very interesting thing. But you wrote in your slide, it was that using this deep learning to recommend what type of measures to use, etc., etc. This is very interesting, but I cannot imagine. You see, for instance, I have some data, and I would like to use appropriate clustering algorithm, and analyzing data, how it is possible. how this system might recommend me what kind of algorithm to use.
Yeah, I will try to explain briefly how it works, the system. It's just our ongoing work. We started to work on the project this year. First of all, any deep learning system or any system based on artificial intelligence first should have some historical data. We should first learn and learn on some examples and then we should apply. We should test on it. I will just briefly present the algorithms that we used. We used some classical algorithms like k-means and k-medians. They are among the most popular that are based on Gaussian clustering, the spectral clustering algorithms, some tree-like algorithms that use hierarchical clustering, like word algorithm, and agglomerative clustering, also Birch, and also three algorithms which are now very, very popular also is DBScan, HDBScan, and optics that use that are appropriate for circle clustering. And also we have Gaussian mixture algorithms, which is based on distributions that assumes that the data within one particular clusters are distributed according to a certain distribution. And those other data, those were our competing methods, because there are much more methods, but we selected these 10 methods to recommend them. We had to recommend one of these methods, but in fact our system may recommend several methods for a given data set. And after it, we used some programs known in the literature that allow us to simulate, to create some artificial data sets that we use for training. For example, we use the paper of César Comín with the alpha parameters to create some clusters with some overlap and without it. Also, we found this popular repliClust software that also allowed us to generate some clustering-related data sets with some overlap or not. And we also generated some Moondate and Circle data sets that we have just seen. And we put everything together. In total, we had 38,000 data sets with different number of clusters, different number of features, and different number of points per cluster. It was using for training. Also, here is a diagram that shows the number of recommendations by when the one algorithm, as I said, ideally the system had to recommend just one algorithm, but in some cases it recommended 2, 3, or several algorithms. Here is a number of cases depending on the number of data sets. We adjusted the parameters, so we optimized the parameters of all data sets. For example, for k-means or k-medians, we just used 100 random starts. It may not be enough, but we tried to give some fair conditions to all competing algorithms. Here are an example of application of results of application of clustering on these 10 different algorithms, and what are the kinds of clusters that can be created using them. What I should mention is that for some data sets like k-means and k-medians, we needed to indicate the number of clusters. But some of data sets like DBScan, HDBScan doesn't need it. The method determines by itself the number of clusters. And we use different kinds of... Yes?
Можно через 20 минут вы мне позвоните? Да, спасибо большое, извините, ради бога.
Okay, so I will continue. There are 3 types of deep learning networks that we used. First is CNN, FNN, where the propagation is almost only forward, and an RNN we used for clustering. And finally, we determined that the best model architecture that provided the best result during the test was this one. It was a CNN with a residual block and self-attention mechanism, but the kind of CNN deep learning network. It was not very complicated, but it was fast enough to do both training and test. the first thing that we had to do is to make sure that there is no overfitting in the data and to select the optimal number of epochs or number of iterations for training. So it has been done also. And the metrics that we used to measure our performances were average loss, the F1 score, and the Hamming distance. Here we have the results in confusion matrices for the deep learning models, so that for each of the algorithms. Here we can see that the spectral, I think the word method and the spectral clustering provided the best results, but we shouldn't compare the algorithms here. It provides the best result for our selection of data set. Here I give the formula of some well-known clustering validation indices with which we compare the results of our recommendation system. Is first of all Silhouette, Karinsky-Arabats, Davis-Baldin, and done in the index. So these indices usually are recommended in the literature in order to select the optimal number of clusters first and to select the most appropriate clustering algorithm for a given dataset. And the first results we obtained were very encouraging. Our model managed to beat the competing indices according to both F1 score and the Hamming distance. We can see the results here. The results of our model is blue. So it's by a good margin that we have 20 or 25% in most of the cases that we managed to gain compared to the competing indices. And very recently, just last week, we applied this algorithm to the real data set because it's almost plausible to apply the new algorithms, new methods on a real data set. And also we obtained some promising results. Here we selected 10 real data sets from the UCI machine learning repository that contains hundreds of real data sets that are used for machine learning experiments. And here we can see here on the left part, our model also provided the highest median and the much better indices, especially compared to Silhouette and Karinsky-Arabats that are usually recommended in the literature as the best selection methods for selecting the number of clusters. So this is I would like to finish my presentation with this slide. Thank you for your attention, and please don't hesitate if you have questions. Please ask.
Volodya, thank you very much. Bravo. You see, this last part, it was really so impressive. The first one also was, but it's something. Okay, questions, comments, remarks, please go ahead.
I have two comments.
Yeah, sure.
I have two comments. First comment is about the picture in the final slide. I consider it's a masterpiece that should not be just thrown out. Not this one, the last slide.
Okay, this one?
Where is this one, this picture.
Okay.
It's a masterpiece. Next, my comment. I think what you are proposing is actually has a potentially may have a big consequences. The thing that I think I had a colleague back then, Fuad knows of him, Ilya Mushnik. He called similar constructions with the Russian word tarataika. And what is lacking now, I think, it should be complemented with a measurement tool for complexity, data complexity. This is, everybody understands that data have different complexities, and there are no proposals in the literature, except in our lab, for measuring the complexity. I think the database that Vladimir has described of, I forgot, 38,000 data sets.
Yeah, exactly.
They generated.
Yes.
They need like a thermometer, not just descriptions, but the complexity.
One more measure.
But the complexity attached to it. Then this can be a very big idea and very much kind of world shaking. I congratulate you, and I think I could help in this. Thank you.
So thank you, Boris. You're always welcome to participate in our research. I will send you the next draft that we will prepare for pattern recognition.
Great. You see, Volodya, when you presented this part, and I thought that the only way to confront it is to create new measures and say that our measures works better, etc. But now Boris already has an answer, proper answer to this situation. I think that you should cooperate and do. It is really, you see, absolutely fantastic idea. Great, really great. Bravo.
But if it works, it will be used throughout the world.
Sure. I understand in that way, you see, in, I don't know, 90% of cases, it will work because, you see, 90% of cases are rather regular in a sense. In 10% of cases, it might not work, but then it's necessary to invite professional people and say that, okay, it doesn't work, what to do. That's a normal thing, you see. Wow. Very, very interesting. Bravo. Okay.
Thank you for your comment.
Concerning this biological part of the story, I have another question, but this question is already, you see, this very article we wrote together with Denis concerning this specialization in the sets of how it's called specialization of cells. Is there, you see, we did it from absolutely different point of view. Is there some kind of genetic basis for this kind of specialization? Do you know such works or maybe ideas to go in this direction?
Not really.
Did you get my point?
Yeah.
I mean that our article, which was published some time ago, you understand, yes? Because
I remember. I had a long discussion with Denis about this article and about possible applications of the method. We may apply some optimization method. We try to minimize or to maximize a certain objective function. But what I said, there are a lot of applications, especially I think in the field of economics that where we can apply this kind of methods. But in the field of bioinformatics, it's hard. It's hard to me to say if there are some extra applications. Maybe so, but I'm not working exactly on this kind of
Volodya, it's not an application. It's a bit different stories. For instance, we showed that how they might specialize due to this and that conditions. But is there, maybe to talk to biologists, to find some species, you see, maybe they make this specialization due to this common, I don't know, you see, some specific genetic features, etc. Is it? I don't know. You see, this biology is not my field, so you're most close of us to this field.
Yeah, I may talk to biologists. I'm also I work in the field of bioinformatics. I'm as you, I'm a specialist in more in mathematics and algorithms. You know, so my knowledge of biology, unfortunately, is very limited.
I see.
But I can talk to show this paper to some biologists with which I collaborate.
Maybe they can find some kind of mechanisms, you see, stemming from biology. I don't know. Okay. Other questions or remarks? You see, I enjoyed this paper very much. Very interesting.
Okay, thank you very much. Thank you very much for your comments. Also, Boris, thank you very much for your suggestions.
Well, I had a number of comments on genomics subjects, but I think that is of interest only to two of us, so I decided to skip it.
Okay, you can write a letter or
that's
talk by Zoom or whatever.
Say of the usefulness of reticulogram or whatever. I don't want to bother.
I believe you have seen it a number of times, but
I'm saying of usefulness of it.
Okay, for the last 20 years, I'm presenting.
Okay, I would like to say that in another network, as I said, well, don't be surprised that only 8 people are here, but in another network, more than 100 people were joined this network.
Perfect.
Okay. So thank you very much then, and see you tomorrow.